This method can help answer key questions in the microbiology field about the dynamics of colonization and infection by microorganisms in the gastrointestinal tract. The main advantages of this technique are that it is more representative of foodborne infections in humans and that it reduces potential tissue damage in fish compared to oral gavage. Visual demonstration of this method is critical, as the washing steps can be difficult to perform because of the highly motile nature of the Paramecium.
From a live growing culture, add 1 milliliter of Paramecium culture and 1 milliliter of an E. coli MG1655 culture to a 10 milliliter tissue culture flask containing 8 milliliters of E3 medium. Lightly swirl the flask before placing it at 22 degrees Celsius, passaging 1 milliliter of the co-culture into a new 10 milliliter tissue culture flask containing 9 milliliters of fresh E3 medium, supplemented with 108 colony-forming units per milliliter of E. coli MG1655 every 2 weeks. To determine the bacterial half-life within a Paramecium, count the number of Paramecia from an optical density of 600 Paramecia bacteria co-culture, and add a 50 microliter aliquot of supernatant to a new 1.5 milliliter microcentrifuge tube every hour for 6 hours. Add 950 microliters of a 1% non-ionic surfactant in PBS to the tubes. And lyse the Paramecia in each sample with 1 minute of vortexing. Then, make 1 in 10 dilutions of each sample in sterile PBS. And plate 100 microliters of each